Mukund, AI on top.
AI. Very good.
I think of biological cybernetics.
AI, cybernetics. I mean, this is an alphabetical order. This is not in any order of this order and that order. So I'll start off with one thing, which is a field which has had a huge engagement of theorists with biologists and is both well regarded and completely core. Nobody will question this particular one. Okay, molecular biology. This is sort of the standard one should be.
I think you can say structural biology there as well.
Okay. So structural biology. Somewhere here. It's part of molecular biology. So okay. Structural biology. Fine. So both things which are sort of. Maybe this is a little less core, but okay. Something here just to get controversy started.
I still didn't follow core versus peripheral.
Okay. So peripheral would be if somebody is studying, let's say, the conductance of electrons in DNA because they're interested in electrons and not on DNA. So it's something which is they're studying a problem which is interesting and probably real good science, but it's not of relevance to how a biologist, to what a biologist needs to know.
So core peripheral as defined by
Again, both these axes as defined by biologists. That's one way to frame ourselves. It's not the only way. Yeah. So something. Anything? Nobody did.
AI has managed to get itself into a lot of trouble.
Okay.
So I think it's managed to get itself close to the bottom there.
Yeah. And the reason? The reason? Did they promise too much?
They promised way too much.
Okay.
And it was a hard problem.
Fine. So
It's still is.
I think it's completely underestimated difficulties with these.
No, but they also overhyped it to a degree which some of the others on the list are also in danger of doing.
yeah. Okay. So similar to the overhyping, I'm going to add here. People who are working on things that are at the core of biology but are in danger of starting to get ignored. Okay. So quantitative biology.
What do you mean by that?
What do you think?
I mean by that, the subject, there are many people who claim to be working on quantitative biology. And there are journals of quantitative biology, and biologists don't read these journals.
But hey, every time I take a measurement, I'm quantitating something.
Absolutely. So this is another question. Why do people get to name the field? But I'm just putting it, yeah, fine. So, but there are people who claim to be working on quantitative biology because they don't want to be seen as working on systems biology.
I think I would put systems
Systems biology on the left.
On the left, systems biology.
Under AI.
Under AI.
Where it should be. Actually, I think you can put it at the top, but I think it's peripheral.
I see.
It's well regarded, but it's completely irrelevant.
Okay, that's interesting.
Yeah, and should not fall with time.
But please comment about quantitative biology applies to systems biology. They're all systems as defined.
No, but so it's not. We're not going to take the most charitable or generous interpretation of the subject. We're going to take the actual practitioners of the subject as they self-define. Okay. So
Wait, let's go back to systems biology. I think it's at least in principle, it's very core in the sense that it's asking how the system as a whole works. Whether it has achieved these aims is a matter of dispute, and of course the name itself is extremely fuzzy.
Mukund, I like to interject.
Yes.
So for those of us who are not really into
very
biology,
very good.
what is the difference?
What is the difference? So, well,
I mean,
fine.
everything looks okay.
Okay, so maybe we can start at the top and give sort of one example of a research problem that would legitimately come out of each one of these fields. So AI, artificial intelligence, everybody is sort of okay with.
Computer vision and stuff like that.
Yeah. Okay. Well, computer vision might fall under computational neuroscience now. But for sure, this is sort of a more legitimate field than AI. Anyway, so bioinformatics, for example, the way the term is used everywhere outside Europe is the study of biological sequences, mostly, and sometimes big biological data sets, but mostly it's sequences. In Europe, it's used as a catch-all for computational biology. Biophysics, we all understand. You take molecules, you pull at them, yeah, that's why, or membranes. Computational biology is currently the least despised term for people who work on systems. This is sort of what people use currently. It doesn't yet have a bad name. Computational neuroscience, things like computer vision, models of the brain. Many people here work on computational neuroscience. Mathematical biology, again, there's a few of these. Mathematical biology, quantitative biology, theoretical biology, all mean roughly the same thing. There are people who publish models or theories of biological systems, but very rarely have an engagement with experimentalists anymore. Theoretical biology used to be absolutely at the core, and now it's not. I don't know why. Structural biology, molecular biology, of course, we all know. Structural biology, similarly, you find the structure of molecules and therefore understand how they work. Systems biology is probably what Sydney is going to tell us more about today and tomorrow. Does that pretty much cover it? Theoretical ecology, similar to theoretical biology.
So population genetics and so on, such.
Population genetics.
That's theoretical ecology.
That is theoretical ecology. I think it's rebranded.
Good. So I think sort of the idea has been, this is sort of work. I think this is what I don't mean to actually try and fill up this. Yeah.
So, why is there no sort of cell biology, developmental biology, that sort of thing here, and why is there molecular biology? Because I think of all of those in sort of core biology.
Yeah. I put it up here because molecular biology, when it was being invented, had a huge engagement with theorists in a way that developmental biology never did.
I see.
So that's why I put it up here.
So basically everybody seems to agree on pretty much everything except systems biology is the only one that has produced any decision at all. So let's forget everything.
Or bioinformatics. And bioinformatics, in my opinion, is actually quite core, and it's getting a pretty good reputation, but people would disagree. So I'm going to leave this. It's probably whetting people's appetites. And the idea now is to maybe give a few examples of the kinds of things that people in this room work on, and then the major question: what is the role of theory in biology? Does it have major contributions to make, and what are the kinds of questions you should be looking at? But we'll get to that. So maybe we can just go along here. So how many people consider themselves to be working on aspects of AI? Zero. Okay, Madhu, good.
Sensory processing.
Okay. See, so this is the thing. Nobody wants to be involved with it, but
It's no different. Are you in it? You want to admit it.
Okay, bioinformatics. I definitely do a lot of sequence analysis and data analysis.
That's a different kind of bioinformatics.
Yeah, no, it's fine. Biophysics.
So is it physics in biology? Is mechanics, for example, biophysics?
Mechanics is not usually called biophysics. Biophysics is, you read biophysics, read the biophysical journal. That pretty much, I know it when I see it. Yeah, that's fine. So we also thought about work on biophysics. Mother is
for future reference. Can you put those numbers there?
oh, numbers.
Who said yes?
Who said yes? Roughly, not who, but numbers. One or two.
Computational biology, use a computer all the time to do model 1, 2, 3, 4, 5, 6, 7. Right? And many of us are raising our hands for many of these things, right? So obviously these are not watertight disciplines. Computational neuroscience, 1, 2, 3, 4, 4. Okay? Mathematical biology. Yeah, so I'm going to put my hand down here. 1, 2, 3, 4. Molecular biology, we have 1.
I mean,
2, 3.
but you mean
And structural.
as a tool or as a question?
Very good. As a question, I mean. As a question. 1, 2, 3, 4. Okay. Quantitative biology. Would you describe at a cocktail party somebody else?
What is quantitative?
Would you tell somebody you work on quantitative biology?
Depends on what you call quantitative biology, right? If I look.
No, but that's the whole point. I think the point is that we keep changing names just to avoid stigma.
But fundamentally, there's many people here who will say no to all of those.
Yeah,
So,
fine.
I mean, feel free to subtract one from everywhere where I said yes. So.
Okay, fine. Yeah, I agree. Structural biology, definitely have a few. 1, 2. Systems biology. We admit. Okay.
This is also defined from a theoretical perspective or a physiological perspective?
Again, would you
I don't know. I mean, the thing is, so a physiologist, and you're a systems
You
biologist.
wouldn't need to call yourself a systems biologist. What's the need?
No, I'm just worried about,
Yeah.
this pegging of things is...
Absolutely.
is-
That's part of the problem. That's part of the problem. So pegging is the issue. Okay. How many people would admit to working on theoretical biology? 1, 2, 3, 4, 5. Okay. How many people have published in Journal of Theoretical Biology? 1, 2, 3. Okay, good. Theoretical ecology. You might be sure that he's out of town.
How many people would admit to working in would not admit to working in theoretical biology but have published in the journal?
How many biologists have read the Journal of Theoretical Biology? 1. Okay, good. Okay, that's sort of a good representation.
But biomechanics is the only thing I've identified medicine for. And that is mechanics in biology. Again, an abuse of the word mechanics in biology because it deals with some narrow power of biology.
Yeah, but it's fine. Okay. It might meld into robotics.
I would call myself a biochemist.
Okay, so now fine. So I think we should stop this particular thread. The point is being made in some sense.
Can I turn this around? So you're asking who's applying theory to biology, but biology brings mathematically interesting problems which mathematics does not know to handle because we know the math of limits very well. But for example, if you take dynamical systems with non-ignorable laws but not swamped by laws, that's stochastic dynamics and there's no sensible general theory. Unlike for dynamical systems or statistical systems.
So you're talking about actual theory where
But I think
filling in the gap.
But theory, as opposed to filling in the gap, biology fills in or identifies giant gaps in our theory and presents apparent solutions which make very interesting theory.
So is that a good place for you to advise students to go working?
I think that's I don't know.
Okay. So this is as much as I planned for the icebreaker. It's a little longer than I thought. So I'm going to ask Sydney to at least say a couple. So for example, ecosystem biology has moved up there. To answer a couple of questions. So one is why do these disciplines keep changing names, although people keep working on the same things? And secondly, if you were to advise students to get into a branch of biology where theory is important, what branch would that be?
I think the role of theories.
Because then we'll get to the big question.
Yeah, I think I would like to hear what people here say more about how they see it embedded. So maybe you should turn it the other way around and ask what are the fundamental problems of biology that require a theoretical framework.
So fundamental problems.
And make it different from everything else.
Yes. Yeah, it shouldn't sort of fall into one of those.
Correct.
Yeah.
And we point out that Sydney didn't raise his hand at all.
No, I didn't raise my hand. I'm too old-fashioned for all of this.
So this is excellent. So presumably all of us think we're working on such problems.
So why do you think living systems are different from anything else? Why is a living system different from a non?
Can you repeat? I didn't
Yeah. So the first question is what are the problems? I've written that. Second question is why is a living system? System is already in that. Why is a living system different from a non-living system?
From a non.
From non-living?
A mountain.
A mountain.
That's what he said.
Yes, it's a good story. Can you do this? That's why I asked it.
So answering this might get you. Fine. Can we?
So what do you think is the difference between a complex system like a mountain and any living system? You see?
Homeostasis.
Homeostasis?
What makes it different?
No, it isn't.
Like static, right? I mean, it's also
The main thing you see is that living systems are the only natural systems that contain an internal description of themselves. There is an internal description of itself. That's very fundamental because Schrödinger made a mistake when he wrote his book, What is Life? And that was only corrected by Feynman later. Okay? So the only living system is an internal description in the form of their genes, their genetic material. Okay, so you can now ask yourself this question, since we can now get access to this complete. Can you predict what the organism looks like by looking from the gene sequence? Right? Because it does contain. So you're asking a question, how are organisms computed in the form of natural computation? This is what I think it is, from their genes. And that's, I think, it's the whole thing. And all of these tackle fringes of this. So, if you like, that is the fundamental problem of biology. And I think you can ask then from any component or any partial, an organ or anything like that, we only ask 3 questions. Okay? We ask, how is it built? So that the problems of construction of this now. Of course, then you want to know how cells are built, how molecules are built, how proteins are read off this. You want to know all of that. You've got to do that. How's it built? And that includes developmental biology. Then you ask, how does it work? What function does it have? So that's the old question, which used to be called physiology. And then you ask, how did it get that way? Evolution. Right? So those are the 4 areas which we need the answers to. Okay. Now, of course, you only need quantitative biology where the amounts are important. Right? And you can go through each of these steps and ask yourself. Now, why I think systems biology isn't the right way of looking at it is, and I will now take what I consider extreme, right-wing, reactionary systems biology. They say the whole is greater than the sum of the parts, and you have emergent phenomena which you can't get by studying the parts. Okay? And therefore, you have to study the system as a whole. Now, that's wrong, because the whole statement is the whole is greater than the sum of the parts studied in isolation. The whole cannot be greater than the sum of the parts and their interactions. Okay? Because that's how the parts, it's the interaction of the parts which compute the whole, just using that in the general thing. We would like to be able to do that. We'd like to be able to say this, of course. So this is where structure comes in. And therefore, we can look at all of these things and say, right, we need to know at each level, this is what we need to know, how the componentry generates the next level. Okay? So we consider a cell. Cell has got hundreds of different kinds of molecules. We need to know how does that, the behavior of the cell, physiology, performance of the cell, how does that depend on its componentry? And similarly, when you take something like the brain, you can now forget about all the molecular biology You don't need that, okay? Because you can reduce this to a new level of units of cells. Okay? It's a network of cells and their interactions. You have to know how the cells are connected. You have to know how they talk to each other. And you can't study that, the whole of that. You have to do it because it's generated in this discrete form. It's generated from a sheet of cells or a tube of cells that are all the same to begin with, and then parts of it change during development. We need to know how that's specified. So I think that we need to know what it is reasonable to ask. See? And why I think systems biology will not work, if I put it that way. First of all, they deny the molecular level. But you see, all of us, all of living matter, is reduced to the molecular level. When we transmit this piece to the next generation, you are reduced to molecular level in the form of the sequence of DNA, plus the initial conditions, of course. So I think you can't ignore that. And the genes control the construction of molecules. You can't ignore that either. And the molecules have various kinds of interactions, some strong, some weak, just like physics. And these interactions will decide on the next level of how those participate in a cell. And we need to have the theory of all of that. We need to have a framework to consider it in. So I think you have to then just look in general terms at any living organism and what we need to put together. So what is the framework? Well, the most important thing is why we find it difficult to do this is our brains act in a serial form. There are only a few things we can hold and work on at any one time. Okay? That also we cannot really comprehend many levels. There are only about 3 levels because of the linear nature of language. So, but biological systems have, in their essence, this vast possibility of parallelism. Bits of it can all be doing their own thing at the same time, and we can't easily comprehend that because we don't think in that way. We don't think with all the parts of our brain working, at least in our consciousness and in our communication. So we have to then look at the power of all of this. So, years ago, I wrote a paper. It's in a very obscure place, but it's a paper that asks the following question. You go to a control engineer and you say, okay, I've got this problem. I've got this chemical factory which has 4,000 different processes running in it, making various componentry. Right? Now, how do I solve that problem? So it's well known when you have such a complexity in control engineering, you've got to have a monitoring system. So the engineer will put a probe in each of the reaction vessels that measure the temperature, the pressure, whatever other parameter, the pH. He'll take that up to a central computer, all right? And then he will have effectors. So if there's a fluctuation, which could destabilize the system so that it either runs to 1, whereupon it explodes, or runs to 0, whereupon it dies. So he knows that, and so he will say, well, open this tap, make it move faster here. Right, now, if you think about the complexity of that computation, you can ask, well, could you actually deal with that in a computer program? And the answer is no, because there are hidden costs here. The computer, because it's a serial processor, okay, also has problems of arranging priorities. So you'll find increasingly the computer will spend more and more of its resources on its own administration. They're called operating systems, okay? And what is left to do the actual work is very small at all. Of course, you didn't tell the engineer, by the way, this entire factory can copy itself in 40 minutes and make another copy. Right, so we have to ask what style? What is the rules now that govern this? Because clearly this model of pipes connecting things would not work in a biological system. We can't implement it this way. So what the system has resorted to, and the key thing is that the components in this don't have to know everything. Nobody knows anything. There is no master computer inside a cell, okay? There is none, okay? So you just know, so let's take one of these little lines in the factory making tryptophan. Okay? So they don't have to know what's happening to histidine or phenylalanine. They're only interested in tryptophan. Right? And the system then does the following, the way it is. It says that these steps here can participate in this way, but we don't have to get the information from some central resource. All they have to know is what is the concentration of tryptophan. All right? And then the system is actually set simply by feedback. So as soon as there's too much of the terminal product, it'll work back on the first step and slow it down. Okay? Whereupon the concentration will fall and the system will regulate itself. And then how is the whole regulated? Simply by participation in global resources. How much energy, how much glucose, how much nitrogen that you have. And of course, in such a system, if any one of these local demons, which you might want to call it, is too greedy, it kills the entire system. Okay. So any system that's too greedy, there will be capital punishment to just be eliminated. Because everything depends on how fast you can grow. So it's self-optimizing, and it's organized in these modules. Now that's what an engineer, not what we expected with this one thing. Now, why does it work in physical terms? Because bacteria are small. All right, now on that scale, you can start to ask yourself, it's about a cubic micron, which is a femtoliter. It's 10 to the minus 15 of a liter. Right? And at that level, the transit time for your average molecule like ATP, molecule of 500 molecular weight, is milliseconds. It takes a molecule a millisecond to cross the bacterium in the average. So 100 molecules is everywhere. So the things are not diffusion limited like this. And then the next thing is it's run by molecular collisions, which are very fast, because most of the time, the molecule is banging on the wrong enzyme, the wrong protein. For the time it bangs on the right protein, it's banging in the wrong place. And the key thing is the active site. the one that has a binding constant. That says you are relevant. Right? So the style of this is, it's a broadcast of information, molecules of many different types, small molecules, moving around very fast. It's ignored by most systems, just go through them, but it's captured by the ones they're relative to. Right? And that's if you like the style. And of course you can ask yourself, well, what happens when we increase the volume of this? If we go to a cell, we've got 1,000 times. We've got 1,000 times the volume. Things wouldn't work. In fact, we know they don't work. So, in fact, you will see solutions of creating compartments in these cells. And in fact, one of the interesting things that can be run essentially on diffusion is, in fact, you can run the synaptic extrusion. It's about a femtoliter. It's a molecule, and you can do everything in that space by just occupying, like E. coli. So it's a miniature E. coli. So that tells you anything at this level, diffusion is good enough, and of course we don't have to do anything special except to describe the relevance for the small molecules of the cell. So that gives us a very good idea of how the thing can evolve. So there's a very interesting experiment one can do, which is quite interesting, and you can test the generality of the control system as follows. What you do is you put the cell in heavy water, and you find out if it can adapt to this. And of course, after a while, E. coli will grow in heavy water, with glucose and so on. And that's just substituting deuterium. It will grow in heavy water at a much slower rate, contains per gene 3 times the amount of RNA that a normal cell has. That means it needs 3 times the number of synthesizing machines and 3 times the amount of protein, okay, that a normal cell has. Now, how do you account for this? Because the effect of deuterium is quite small. You account for it because there are some steps in this pathway in which the isotope effect is enormous. Right? And if you look at any step where you're removing protons or adding protons, like a dehydrogenase, or any step that involves this, you find that the rate of change is, you can look this, people have measured the enzymes, they can be from 11 times slower, not just the fraction you might expect, right? 11 times slower to 40 times slower. Now let's imagine we have a chain of a pathway making tryptophan, and
I think people here may not be able to hear, so maybe that's why I got you this special chair.
Okay.
That's why I got you.
Okay, good. So we've done the experiment. Now what we know is that heavy water did not exist even. If you buy heavy water 99.9% pure, you don't have to use such a high amount. It's cheaper if you do this. Heavy water does not exist in that form anywhere in the universe until, I think it was 1936, where it was made in Chicago for the first time. So E. coli cannot have a memory of being living in heavy water. So no living organism can have those. So this means to these completely strange things, it now can adapt. It can actually change everything to do it. So let's just see one case. I started with tryptophan, but I prefer to use, say, uridine, which is a component of the hormone. So there is a step in this pathway which involves a dehydrogenase. So everything has been selected for the amount of protein, because what you have to realize is protein synthesis for bacteria is the biggest constraint they must manage, because it's the most expensive. That's why so much of the machinery itself is devoted to making proteins. Right, so the first thing we do is we put it in heavy water, and because there's this dehydrogenase, the amount of the terminal product, which is UTP, falls. At that point, the enzyme system will be automatically opened up. More will be pushed through. And if that's not enough, there's an automatic step to make more enzyme. But unfortunately for E. coli, the enzymes are coded in units, in which all the enzymes of a biosynthetic step are clustered, called an operon. And so E. coli cannot manage to grow in this. E. coli cannot manage to do this without switching everybody up. So to get, let us say, 11 times more, even at maximum transit through the system, of one protein has to make 11 times more of others, which is not necessary. And then, of course, we can calculate the excess of protein, because having to make more protein, so it's a tax. Now, and we know what, we can measure that tax in absolute terms quite easily. So we give it tryptophan. We just give it free. And by the way, it doesn't matter whether you give it protonated or deuterated tryptophan. It makes no difference. And you give it tryptophan, and what has happened, you'll cut the demand for this step. So we can measure exactly how much and it will fall to zero. You don't have to make them themselves anymore if you get a free lunch. Because that's how it all evolved. It evolved in bacteria because they came to live inside us or on us. There's an opportunity to be parasites. And so you needed a thing which you could switch off when you got out into the cold world outside, where there was no future to think, but when you were inside, you could get this free lunch. And the whole of that, and we know that, because we know that all these bacteria have two genomes. The one for basic life, and if you go to bacteria that haven't left the original environment, in fact they still live in boiling water. What you find with these is there are no control mechanisms. There are no pumps to pump protein, to pump amino acids into the cell from low concentration. All of that has been added as a consequence of this parasitism. So, I mean, before there were animals to live in, bacteria had to make their own way. As soon as you could sit on someone else or in someone else, you could get a free ride. And that evolved so you could still retain your ancestral characters and not have to be worried when you get thrown out of the living system, you can't grow anymore. You were committed to parasites. Some of them are. Some of them have gone so far to say, I will get rid of being able to make these amino acids at all, which means when they get thrown out, they can't survive. So I tried to do this to say, see, there's a phenomenon that I have to explain. So it isn't that genes know all about this. I can't explain it and say, well, it's this. We know that everything will be accounted for in molecular terms. We know exactly how these proteins interact with each other. We have to work out how the regulation is achieved by blocking it. And you can make very good analyses of how the system could have evolved. Okay, so I think that needs a theory. It needs a theory, it needs an understanding. What life at essentially what has been called low Reynolds number, what that really means. Okay, that everything can be controlled by molecular confusion, by molecular interactions, molecular fusion. And that makes it quite easy to see, in principle, how things could evolve and how this automatic system contains a generator. So there's no control that is central. There's no master program inside the bacteria that knows how this fixes everything up. It's all done automatically. If the bacteria in one system becomes too greedy, then the system will just be eliminated. And I think that is the kind of being spelled out. Now the interesting question is the following. So here's an interesting question. If I gave you all the information, namely what was the affinity constant, what was the molecular rates of transformation, which it would involve knowing the activation energy of all of these things, could you compute the behavior of E. coli? Could you predict how fast it will grow? Now that is the thing. That is, I've now spelled out what I call the wiring diagram myself. So all these autonomous things, all their stops and so on. And now the problem is, can you compute how the cell behaves? Alright? Now it doesn't do it by a lot of partial differential equations. It doesn't do it. Because that's more. So it does it because it has these discontinuous steps which respond in that way, and you have to know what those parameters mean. And of course you can ask yourself, if I change this affinity a little bit, what happens? And that can be done as an experiment. Now, so there are people, a man called Tolson in San Diego, who is a chemical engineer by training, by the way. Engineers know all about this. And who has got what he calls systems biology, but I don't. See, because systems biology would say, we'll just measure everything. We'll measure all these things, plus what are the important interactions in this. We won't be able to predict sudden changes.
So, Sydney, just going to interrupt you, since you had that question, I just want to get people's opinion on the answer to the question.
I've given you this, because I think all of this follows. You can take this to the next level and think about it in the same way. So I just don't think that, I think all of these are just clicks. See, we probably have a quantitative biology because a group of people got rejected from the systems biology journal and decided to found their own way. Although I will tell you that the Cold Spring Harbor Symposia, which are now more than 70 years old, are called the Cold Spring Harbor Symposia in quantitative biology. You just think about that. Very old. Right, sorry.
No. So I thought it was fairly coherent and it sets everything up. But I know that there are some disagreements here. So let's deal with the last question first. So first of all, given all molecular interactions, affinity, mass distribution constants and so on, can you predict how fast E. coli will grow? And second question, is that the right kind of science to be doing? So,
I was just wondering, so there's one question which is E. coli grow or not grow. But I was just wondering in terms of, if I have a group of cells, then the whole organism might want to take a decision, right? And I'm just wondering in terms, so one thing is to explain one cell, which is, and one cell's decision to do X or Y, which is a binary choice or whatever, right? But I jumped on C. elegans, which was a beautiful thing of combination of multicellularity and simplicity, and it being able to take, and having very simple behavior, but able to take a larger number of decisions. Like,
yes, but you see...
And how do you have a group of neurons, a group of cells, which decide what, I mean, if I have a group of decisions awaiting me, how do I compute what decision to take, right? And I think that is...
Yeah, I think you have to follow this through for everything. I'm looking at E. coli. If I want to know how the culture of E. coli grows, I have to take in consideration the interaction between the cells. And we know that signals pass between them. We know that they can cluster, can aggregate, etc., etc. And I think you have to go stepwise through the evidence. Right? So for you, I'm not saying that you should know all the rate constants for the potassium channels in those neurons. Consider the neuron, consider its output, which is you can get rid of what someone else would have done before and tell you to have this potassium channel, you'll get this resting potential, it will be automatically rectified. That's all you have to know. Someone will calculate it all. That is the person at the level below. So now you have neurons and they can send signals to each other, they can interact, they can make a decision. That's your job. You don't have to worry about all of the other stuff. Someone else will do it. So, if you like, this is pragmatic reductionism. You reduce it to the level of units that you need in order to explain your phenomena. We don't have to stop with the hydrogen atom. So quantum mechanics is completely useless. And we're not interested. Someone else. And I remember, let me give an illustration. I was once a professor in a biochemistry department, a visiting professor at Cornell. And they had a very big group of people looking at enzyme mechanism. They had a very good group of people looking at the behavior of molecules and so on. And each one was disappointed in the lecture. So the biochemist gave a lecture. They didn't talk about orbitals, and they didn't talk about the level of theoretical chemistry. And when the physicists gave lectures, the theoretical chemists were disappointed. Because here this one man gave a whole lecture on one reaction, which is sodium plus sodium gives rise to Na2. And he was considering just the physics. So each one uses their own units. Now one of the things in biology is you should have enough understanding of the lower levels. So at least you can see the impact. And so I think that's what the nature of the theory will be. That you have to have the way it's organized and how it's put together. I remember I went to visit Jack Cowan Jack Cowan was a theoretical physicist in Chicago who moved to the nervous system. He handed me a piece of paper which was written a very large integral differential equation. And he said, that's the way your brain works. And I said, Jack, maybe the way your brain works, but I know for certain my brain doesn't work that way. So I think it's just a difference. And I think you see what you can't escape from. All the systems biologists want to escape from is this fundamental property of living organisms, which they hand a description of themselves to the next generation. And we have the job now of finding how is it made explicit. How is it made explicit? So that's our job in biology. And it's all based on the script.
So I know for a fact that many people in this room who do things which they would call systems biology would not tend to work at the kind of caricature of systems biology that you've described. But there's another point also, which maybe somebody would like to jump in. There are other, so even if people accept all these pieces of your prescription, the nature of the important problem, the fact that you have to look at how it got that way and so on, you might still want to make theories which are not underpinned by a molecular level of description, but something which deals at the level of phenomena. And again, I know many people in this room work on things like that, and things that have been quite successful in physics. So, does anybody here work on something of that kind, where you work on theory and start coming up at the level of molecules, interactions, building up at that level? I do, yeah. So, would you like to?
The way I understood the three things, how is it built, how does it function? To me, how is it built in physiology? How does it function in behavior? And why is it the way it is?
No.
So
how it's built used to be called anatomy, basically. It's just the construction of it. And how the work is physiology. Physiology is the study of function.
So that's where I think I beg to differ. And the reason I differ is you started with saying there's an intrinsic code, the intrinsic code leads to how it is built and that leads to how it functions. And there's a wrapping around question of why did it come to be the way it is. So why did it come to be the way it is, to me, is to a very good degree a function of its interaction with the environment. The interaction is super important.
Sure, it's evolution. So that you have to do it. That's another subject.
So
I don't care what you call it. Evolution implies that things have selective values, and I just see you can study it. It's sometimes called population biology, or ecology, but you need to make a study of that yourself. What I object to is the following. And let's just look at this. We take a simple model here. Okay? There's a man playing a drum next door. Okay? And we wired it out, have microphones leading here, and we are making measurements. Making as many measurements as possible. And we're measuring the sound emitted from that into ask. We're going to ask the inverse question. We're going to ask, from the sound, can we predict the physical properties of the drum? All the properties of elasticity and so on. Now, that inverse question can't be solved as such, because information has been lost. Like in crystallography, you lose space information. So the idea you could just solve it that way, which people believed you could do, was knocked on the head. You need either an a priori model, you need a priori information. And you need to have some way, because you don't know the distribution of molecules. That itself is a variable. and how can models, I should say. You either need that, or you need to get the information in a different way. Because what we know is if we had the structure, we could predict the X-ray diagram. And if we studied the drum, we could compute the sound it would make. So the forward question is in many cases soluble, and the inverse question is in many cases not soluble, but only in a very limited region of solutions where in fact linearity comes up. Non-linear things just kill the solution of the inverse question. So I think that is what the systems biologists, of course, many of them are compromisers. They're not real true conservators. They'll say, well, you've got to reiterate, you have to perturb the system and do more experiments. And basically, they find themselves solving a whole lot of forward questions. But it is not enough to record the entire behavior and believe you can find a model for the inside, which is what most of them really believe.
Right. I was fascinated by your example of the control engineer, and I was wondering how that relates to, let's say, solving an E. coli problem. Given all the interactions and you want to predict how it grows, given that it's a living system, whereas a control for a chemical plant is non-living, and you have all the interactions and a control engineer.
That is why you have to really be very careful how you look at the system. So let's just say we have, like a cell in our body that has 20,000 proteins. So we draw a matrix. So 20,000 proteins, 400 million interactions. Now, if we assume that is what is going on and we have to account for all of this, it's just noise. Okay? It's like saying we can learn a lot if we have all the interactions on Facebook. It's 100 million people interacting. Because if you do not have clumping, that is, you do not have a sparse matrix, that is what I call strong interaction. And what do you find, of course, is that there are strong interactions. You find that no protein ever acts on its own. It's always organized into an aggregate, and you can now study the details of these things. We don't know too many of them in enough detail. And we find, that on average, if you give me 10 is the average number of components of a device, then I've reduced the 20,000 by 20,000 to 2,000 by 2,000 devices, molecular devices. Now, I then notice that the cell is big and that not every part of it is homogeneous. That things are in certain compartments, what's in the nucleus is different. So give me 10 compartments. So I've now reduced the problem to a small interacting system where I have a couple of hundred things in a membrane, or a couple of hundred different things inside a nucleus to deal with. So the module, sometimes called channeling and sometimes called modules, that is essential to trying to understand these things. It's absolutely essential. And you must have it. Now, the other thing that's missing from people's concept of this is the question of time. So if you take the entire field of signal transduction, they are worried about crosstalk. Right? Now, what we do know is that crosstalk is not a problem. Okay? Because of the way it's organized, the crosstalk is simply reducing concentration to a very low noise level. In fact, if you study the interaction of these proteins at the level of concentration that they actually are available to each other on this, you'll find you get no reaction. Okay, it doesn't work. So people, in order to find the interaction, do them at 100 times the concentration. Then they start to worry about crosstalk. So it is all carefully traveled, and it's done so that pieces come together. There's a communication system, right? And you do it. Now, let's take a following example, which is good. Man comes to me from a pharmaceutical company, and he says, I've got this drug, its affinity for the beta adrenergic receptor, I give you all of this, we've measured it. He says, tell me what it'll do to the electrical phenomena of the heart. Right? Now, we can't do that now because we don't have the right information. So therefore we have to structure our answer in order to do this. So in outline, here's what we have. We know that this adrenergic receptor interacts with another protein, okay, that protein then activates another part of the membrane, activates an enzyme that makes cyclic AMP. Cyclic AMP is a small molecule, so it can diffuse, right? And that near these receptors, there is this incredible unit which consists of a protein called an anchor protein, and it's bound to something called the ryanodine receptor, and on this there is an enzyme which, when the cyclic AMP, it's activated, and it then goes to the receptor and changes a phosphate on it. Okay, adds a phosphate, and the channel opens, and calcium comes out. Right? So we do all of this, and we can now look at the details of this. What we find is that there are lots of other gadgets in here which have the following function. First of all, we are amazed to find an enzyme which destroys cyclic AMP. So that's, say, why should you have an enzyme to defeat this? But we now find that when it's bound to the anchor center, it has a very low activity. Very low activity. So one of the functions of the activating, side functions of the activating enzyme, or once it's activated, is to go to this enzyme, phosphorylate it, right? And its activity rises and it destroys cyclic AMP. These are well-known synapses this way, in which you take a wave of concentration, and you chop it. You chop it to a delta function, essentially. Because once it's done the triggering, it won't work anymore on that system. The system is closed. Right? Then, at the end of opening this, we're going to close the channel. So what you find is that there are a whole lot of enzymes which need to restore the ground state. So, if you like, this could be considered as an oscillator. Right? And we would like to know what is the period of this. Because what we've done is we've converted, through these molecular interactions, we've converted the occupancy of the receptor ultimately to the ejection of a toxin calcium. And at this stage we can say, look, we can forget about molecular biology, we can now describe the system's parameters of this. It will be how many of these are in the cell, where are they in the cell. It's good to know how they work, because then we understand. But we can think of this and move to another language. We move to device language, which we can represent either as a mathematical equation, or if you like, as a quasi-electronic circuit. We can say that this is the output. Then we'll go to the next stage and ask what the calcium. So all of the signaling must have machinery which restores it to the right state. And understanding that becomes an important part, because we could kill that. For example, we can predict that if we modify this receptor at a certain point, so you couldn't phosphorylate it, the system would go into spasm. Okay, because the cyclic AMP wouldn't be destroyed. Okay? And there's no way of destroying it because this enzyme couldn't be activated. Or we can change it in such a way that we keep the enzyme always activated. So the system would die. Okay? That is, all cyclic AMP would be destroyed before it got there. So you have to know all of that, but then I agree, at the next level, we simply consider that. We then ask ourselves, what is the fate of the calcium? We know calcium goes, it will activate the heart muscle, it will do some very interesting things in the special parts of the heart, which have some other gadgets in it, and therefore it will slow the heart. So I think, or speed it up, depending on whether it's an inhibitor or an activator. So we could compute it simply in device language without any of this molecule. So I think that's the big job we have to do. We have to find out where things are in the cell, what do they do. See, and you can't explain on ordinary physical-chemical principles, you can't explain how your cells measure the concentration of glucose in the blood. Why? Because the interaction of insulin, which is the regulating molecule, with its receptor is essentially irreversible, has no dissociation constant. So what you find is you couldn't do that because the margin is. So bacteria, your cells resort to counting. All they ask is how many receptors have been activated. And they count this by a special device of binding an enzyme to the activated receptor and activating it, so it produces a small molecule, an inositol product, at a constant rate. The rate depending on how many receptors have been activated. And therefore, in the absence of that, so it's not meant repairs, it is a counting mechanism. And still very good at counting molecular events and converting them into analog signals. So you have to know all of that in detail before you can begin to understand how this works. And I think that should be the program of everybody. We need to know what are all the devices in the cell, where are they? Because sometimes the only way you can restore the active state is to destroy the activated receptor and make a new one. Okay? So we need to know all of that before we can begin to compute behavior. It's not just a matter of doing the arithmetic and just adding everything. So I think there is a real program for systems biology, but they won't do it because it's too difficult. It's very difficult. very difficult to measure these time constants without perturbing the system terribly. So I think we have to await that.
I was hoping.
No, I think that actually what you are describing is very much along the lines of what many systems biologists would accept the label, think is the way to go.
Yeah, but they don't accept it for themselves.
Very well. Anyway, so I would like to bring up another point which you had mentioned earlier, which was pragmatic reductionism.
Yeah.
And so I'm interested in memory.
Yeah.
And I think this is one of those problems where we'd love to do reductionism and abstract out the gory details of what's happening at the molecular level if we could. But it's extraordinarily difficult to do this. We already know from experiments and models too that you have this horrendous or wonderful, depending on your perspective, interplay between what's happening at the electrical level, what's happening between small parts of the cell, electrically speaking and chemically speaking, what's happening with the genes which are turned on and off by electrical inputs, and of course then what's happening in the vicinity of the cell by way of diffusion of molecules, and of course then the network of the cells itself comes in. All of these are ingredients in memory, and all of these seem to couple to each other very tightly. And it would be nice to be able to abstract out some of these things, but I think it isn't working.
No, I think I disagree with you. If you're interested in memory, I think there'll be other people who are interested in, we have a general theory of the correlation of semantic activity, correlation of stimuli, and in storage. And of course it's stored in different parts of the brain, so you have to do that. But you can study it, you can simply say, oh, there will be a mechanism. And for people who keep on thinking of molecular engrams, the basis for memory, and it has to be coupled. There is a necessity to have that in nervous systems. You need this coupling of the activity, the function, because it's not completely explicit. We know that as the nervous system gets more sophisticated, you don't encode all the pathways there. You may see other games. Then there's the DNA engram, but it's a population memory, and it's in the form of how to build a nervous system that performs those functions. But for us and mice, and I think probably even at some level parts of flies, for these organisms, you have to match performance to the environment. So you have to have a process which is, if you like, learns, and does that. And we know that exists, and it is only very recently, in my opinion, that people have begun to uncover the essential features of it, which involve, it looks like, but the synapses you will retain are the ones that let calcium into it. So you need to have the entry of calcium. Okay, and of course you need to have the entry of potassium or chloride in order to achieve functions in that cell. But in order to preserve and learn and to do the correlation between activities, we need to produce another subunit which goes into these receptors and which is induced through an entirely new system which is activated by intracellular or intranuclear calcium calcium is the important thing. And you can study this. You can study this, actually, you don't even need neurons to study it. You can study it in vitro. Very easily. In fact, it's what happens to your muscle when you train. And for many years, people would cross one nerve. Their muscles that can contract very rapidly, called fast muscles, and muscles that contract very slowly but can exert strong force. They are called slow. And what happens is they crossed a nerve that came from, that was on a slow muscle, they put it onto a fast muscle, and vice versa, and the muscles went according to the nerve. The old experiment was done in the 60s and 70s. And for years, the people worked on this. They looked for factors that would specify the coming from the nerve. They didn't find it. And then someone showed it's just the electricity. If I do that experiment, that I stimulate fast muscle with a train of electrical impulses that have the pattern of slow muscle things, I turn the muscle into a fast or slow muscle. Right? Now, you can actually do that in tissue culture. So I think that's fantastic, because now I can study in great detail how this is achieved. Because we know lots of things change. You need different myosin, you need different troponins, all the components. In fact, it is now becoming pretty general that when you start to make the specialized cells in vitro, make heart muscle cells out of fibroblasts, you need to put them to work. You've got to contract them and let them work in a way, otherwise they won't achieve the correct physiology. So for all the systems, and I think the nervous system is the most important one, in this, there is this entry of the environment so that the performance can be matched to the demands that you've done. So you can change the muscles according to the electrical impulses that of course come from your brain. And I think that is going to be essential in understanding. I mean, now people have found the same thing in the cerebellum, the hippocampus. We know you need this in order that you can have the things accord to the environment. So that is why the connectomics so forth of the higher nervous system will be incorrect, because if people are busy counting all the synapses of all the neurons, which they can only do for a small subset, you have to ask them saying, okay, I'll take another mouse with the same genetic constitution, cannot find the same Wadia. Or I'll take this mouse and kill it 20 seconds earlier. Will I have the same Wadia? Answer is probably no, because it's a dynamic structure and it's changing according to the inputs. So I feel that that's going to need much more than, the sort of hardwired clockwork. Wound it up and now it'll just go on ticking. It needs interaction.
Well, I completely agree. I mean, I think it's very well established that all the higher organisms have very different brains which end up doing the same kinds of things. And so mouse A and mouse B, or for the matter, rat A and rat B can learn the same thing.
Yeah.
And one of the questions in memory, which is one of the things I'm very interested in, is how do you store the same information in so many different ways?
That I think, you see, we need to do something which I think answers another question. Okay, people can put electrodes into certain neurons, and you can show the person, the monkey, you can show the picture of Marilyn Monroe. And there are some neurons that will respond to Marilyn Monroe. Now, of course, what we then say, oh, this can't be the case. So do we have a neuron called the grandfather neuron for every person we know? Right? And then, of course, you can find out that there are some neurons that respond to Marilyn Monroe, just the color of her hair. I mean, you can do this. But what we can't answer, unfortunately, is if I had an electrode in every neuron in the brain and showed the person Marilyn Monroe, how many would respond? See, because I think the information is distributed. I don't think it's canalized into one thing. Because I think what these neurons are responding to some feature of the picture and then send that semi-digested information further up. And I think then the brain recognizes that this is indeed Marilyn Monroe, but I think the activity is everywhere. The activity is not everywhere, but it will be quite a lot of neurons involved. So of course, if you damaged any one of them, it's still recognized Marilyn Monroe. You wouldn't find her with a hole in her face where the neuron was missing. So I think it has to have that kind of distributed activity. And we know that many cases from your own way that you sometimes, I mean, I can give you a very good example. Somebody once asked me, who discovered interferon? And I couldn't remember at the time. And I then said, it was E.P. Abraham. And then said, no, he discovered cephalosporin. And then it took me 4 hours. And I suddenly said, no, I've now remembered the name. The name was IISc. Now, for those who read the Bible, Isaac and Abraham had a very interesting relationship. I was drilled in this when I was a kid. So I had to use that connection that Isaac was his family. Abraham, of course, God asked Abraham to sacrifice. But I had to use that connection to actually close the gap. So I must have been running around in my brain, well, for 4 hours, and somebody came back to me. This little connection installed a long time ago in a biblical story was the one where I could then jump and then reconstitute the entire picture. So, and I think if you do that, you find that these missing names can sometimes reappear, but by ridiculous connections like this, and they take time to compute. That's the interesting thing. It took time to compute. I went through in my mind, imagined the paper, tried to imagine people working on interferon, and suddenly it came to me through this connection. Just the names. It's interesting.
But you were not doing it consciously, right?
Well, I was working on my brain. My brain was working,
Your brain
trying
was
to
working.
recall it. But when it emerged, it just emerged like this. And I suddenly realized, yeah, that's probably how I made the conclusion. It's probably how I did it the first time. Because these were real names of people who just coincidentally had that. So I think it's an interesting thing.
So I'm going to start winding things down so we can wait for lunch at 1. So, last couple of questions and then maybe closing comments from people who haven't said anything. Yeah.
So just so it turns out that theory and feasibility meet played a fundamental role in development of physics.
Yeah.
So from what I understand, what you said, you're partly skeptical about this because theory, fundamentally in biology, it has this inverse problem issue. You see that maybe so you have the construct theories that essentially are not tied to this sort of problem, the inverse problem, and then
I think it is the theory of computation that will do this. Because of course, I believe that there is this in which activity is done. Now, of course, physicists have got a language, it's called mathematics, and in fact, I can't understand many mathematics, except if I think when I see the integral sign, I say the integral sign, I have to do it by pieces, I have to do it discretely, by discrete summation. And the reason is that I think in terms of discrete computable steps. And so my idea is, the integral sign is in fact a program that you put into this incredible mythical computer, okay, and it says do it all together at the same time. I have to do it stepwise. And I think that's what it will be, I think, discrete computations in this form. You won't be able to write equations, except of course when it is the same. You have to know, so it will be very much, there will be critical phases, there will be phase changes in the system. So it probably has to fit that.
Make a point. I'm not asking you a question. So, all of this research on your list are, what is fundamental biology and what physics can be done. There are also some people who very strongly believe that there is no physics to be found in biology.
Well, for example,
Would you like to, some people believe that. So, would you like to, physicists like to comment or you would like to comment on this?
new physics to be found in
Well, that is what people thought, you see. Max Delbruck thought. In fact, a lot of the people thought there would be new physics, new physical laws, okay? And it's nonsense. It is not, you see.
No, but this is, well, that's clear, but something slightly less grand, which is maybe Manu, you can describe the kind of thing you said earlier. A new mathematical.
Or in biomechanics, for instance.
Yeah.
So, I don't know about new physics, but mathematical problems which we don't know how to solve exist all the time in biology. And one example I think was large noise in dynamical systems. And that, in some examples, has driven new mathematics.
Yes, I agree with you. And the examples of inverse questions have driven new mathematics, new ideas about probability distributions. They've driven all of that. So I think the tools of mathematics, will be very important. And I think that I think you see that these. See, you can ask yourself the following. One stood in a boat and looked at a marine diesel engine. I don't know if you've ever seen this. Now, how these things moving up and down at different rates, things opening, closing. And of course, you can't understand that machine at all, because what's hidden from you is the crankshaft. The crankshaft that is driven by these pistons and cylinders. If you don't have that concept, so I think we have to have these fundamental descriptions of the form, which we like the style of the system. This is one which is diffusion control. This is one in which you have really important transport mechanism that carry things long distances, you have to solve them. The fundamental problem which physicists have doubted as physics is this thing of the internal description. I felt that that's not enough to have that. Now, why was that so? I thought there'd be new laws of physics like complementarity, which Dilbrook was very much in favor of. And there is part of biology that no one's ever doubted was just physics. Fluid dynamics you can study in both capillaries, and there's a lot of this. And as physics, it just is a different kind of pipe. The physical laws will apply. The fact is that all biological things are consistent with the first and second law. The first and second law of thermodynamics. Okay? Which states simply as follows. You get nothing for nothing. It's the first law. You get very little for something. The second law. And the third law just said, try and do it as close to absolute zero as possible. So it's just elementary. But there will be. And we can use, when a system can be seen as a physical-chemical system, of course we will use all the physical behavior here. And of course biological systems will be. But you see, we introduced a completely new characteristic into physics, which is the concept of information. Nobody thought that information could be quantitated and physically explicit, made physically explicit. It's sequence information. But that was completely new.
Yeah, but it's not in terms of a grand new physics theory, but in the collective behavior of particles which are subject to constant inputs of energy has opened up a new branch in physics.
Yeah.
And this has great relevance to cytoskeleton in the cell, how the active processes happen in the cell.
Yeah, please do it.
Yes.
Please do it.
The point is that this seems to be the natural language to put in things like cellular regulations. You put in more information regarding what regulates the system. This gives you the natural framework for it.
Yeah.
And right now there are people trying to marry this kind of active cellular mechanics with how molecules are driven, how information is processed and managed in the cell.
Sure. I'm not saying, I don't care what you call it, it's parts of the story. And I think everybody will have a page, or at least a footnote in the final book that will be written. And I think we need all of that, because let me finish with one generality, which I think is important. And I only came to this conclusion quite recently, and I put it in editorial, Terry Schmutzky and I read it inside. See, I used to think there was data, and then there was knowledge, and that knowledge required theory, how to use the data. But I think there's a third way, called understanding. Now, let me give you the concrete example. The annals of the world, the data was collected over a long time. People who described these animals and showed them. Linnaeus turned that into knowledge with his classification. But to understand what it was all about, you had to wait for the theory of evolution and the rise of genetics. Without that, you don't understand how all these animals came to be. And that, I think, is we require, I think, much the same. We have to understand where is the molecular thing directly relevant, and what are the things where we can take the behavior of the system below and convert that behavior up to the next stage. And I think that that's the only way to do it. We won't have grand unified things, that's for certain. Except this one, the internal description. This makes us different. Now finally tell you the story of the Mount, what I said about it. I went long ago to Japan and we were exposed to a very long lecture by a Buddhist in Japanese with a translation. The lecture took 2.5 hours. At the end of this, somebody asked the Buddhist, he was waiting to receive questions, and they asked him, what is the Buddhist definition of life? And he said some Buddhists believe everything is living. Mountains are living. And then I stopped him. I said, mountains are not alive. And he said, how do you know? I said, you can't clone a mountain. It's got no internal description. You clone everything that's living, but you can't clone a mountain. Mountain is decided by the interaction of physical laws, and it is a phenomenon. And I think there are some very interesting questions that you have to ask in this, and I think that that's really going to be, it's going to be the very, we're just going to open up the whole of biology to a completely new approach. And all these little warring tribes will disappear.
On that good note, next is me for a marathon. And I apologize to anybody I offended my little game at the beginning. We'll have more discussions continuing informally, and we'll spend the beginning.
I just wanted to thank Mukund for